Okay, in these lessons, we're going to look at this electron microscope and the principle of electron microscope. You just need a little bit detail only, no need to go through the principle or memorize the principle or remember the principle. Okay, because there's no question I'm going to ask you guys about the principle. Just need to know, because later we need to identify which one is the TEM, which one is SEM. Okay, TEM stands for transmission electron microscope. SEM is the scanning electron microscope. So as I said that, if you to understand this, the electron microscope, always remember they use a beam of the accelerated electron, not electrons. Okay, electrons have no, you can't use the electron, you must accelerate them. Okay, you give them sufficient energy so that they are ejected out from the shells. Okay, so electrons microscope, as I said, that they are about 1,000 times shorter compared to the visible light photons. So means that they have the higher resolving power. Okay, always remember when the wavelength shorter, so increase the resolution. Okay, so we have two types of the electron microscope. One we call as a transmission electron microscope, known as TEM, and we have scanning electron microscope, known as SEM. So if you look at the figure here, the picture here, this is the ultra structure, 2D basically. And this one you can see that it scans the surface, so it gives us a 3D image. Okay. So if you view it this way, then you realize that if you have gone through your practical previously, okay, in your high school, you know that the size of the microscope, right? Okay, roughly about the height of one monitor, something like this. Okay, not so tall. But if you compare the electron microscope, this is the table height. So you put another table, so it means that it's very bulky type. So it's very, very bulky type, and it requires clean room to do this. Clean room to do this. Okay. So what's the principle of the electron microscope? You look at the transmission electron microscope compared to the scanning electron microscope. So you can see that this is electron guns that produce or eject the electron out, and then we need to focus this electron beam onto, okay, electron beam highlights onto what? Onto a specimen. So if you want to focus them onto the specimen, we need to have the condenser. Same thing, we call them condenser, but they are not using the lens condenser. Not like the microscope that we use the lens condenser, we use the electromagnet, okay, to focus them. View it, look at this, where we put the specimen. For TEM, from the word transmission, we want the electron to transmit, pass through my specimen. Can you see that? The electron, we want them to transmit, pass through. So this is where we place our specimen. Okay, so when the electron pass through the specimen, then we focus them onto the screen, then we can view it. So this screen is not a normal screen, but it's what we call this something like the films, okay, that allow us to see the image, and the image will be black and white, or called grayscale. Okay, why we use a grayscale? Not the color one, because they are not visible light. Only visible light give us color, but electron won't give us color. So when the electron hits on the screen, it will produce a signal. So this signal will be captured here. Okay, so how to prepare a specimen, I will talk about it later. It's not a simple process. How about scanning electron microscope? Scanning electron microscope, generally, we want to see the surface. So we place the specimen here. So let the electron actually hit on the specimen, generate what we call the secondary electron. So these secondary electrons will be detected by electron detector here and give us the image. Okay, so for both processors here, for both type of electrons microscope, the specimen here, you have to be careful. We cannot use a living specimen. Okay, why we cannot use a living specimen? Because any living specimen, we put it inside, it will be dead. Why? You try to imagine I put you inside and you are bombarded by electrons. You will die. Are you clear? Okay, and electron is very, very high energy. So it means that the specimen here must be treated with heavy metal. Okay, now this one you have to be careful. Okay, when you process the specimen. Why we need to use a heavy metal? Because we want to create a contrast. different organelles will take up this heavy metal, a different degree of heavy metal. Okay, so let me show you guys this thing. If this is my cell, I use a very simple, the cell and the nucleus. Let's say cytoplasm, nucleus. So if the nucleus take up the what we call this heavy metal more, if the nucleus take up heavy metal more, higher, okay, they absorb higher, cytoplasm lower. So what will happen here is, if this is my specimen, my cell, this is the nucleus. So when electrons bombarded, if the electrons, okay, pass through, transmit. So can you see that more electron will be able to pass through the cytoplasm? Cannot. Can you see that? But less electron, because it's going to be blocked by the heavily stained nucleus, because nucleus take up more heavy metal. You cannot use a difference, a simple dye, because dye won't be able to block or stop the electron. So less electron pass through. So it means that when you look at the screen, you can see the contrast now. Are you clear? You can see the contrast. So part with more electrons will, okay, and part with more a small or lower degree of the electrons. So this is how we actually make the contrast. So we have to stain or treat my specimen with heavy metal. Okay, not a simple process. Okay, but for scanning electron microscope, we're going to coat the cells or coat my specimen with the heavy metal. Most of the time we use gold particle. Okay, we use gold particle to coat the specimen. So what will happen here is, now look at this, because, okay, let's say this is my specimen, okay, the shape here. So we coat it with the gold particle. Okay, so we let the electron bombard it. So when the electron come and bombards my gold, because the electron with high energy, right? So it means that it's going to help to reflect, okay, or generate what we call the secondary electrons. So gold particle, when they receive the higher energy, receive the energy enough already, so it's going to kick out the electron. Okay, it's going to kick out the electron. So this electron will be detected by the electron detector and then generate the image. Okay, so this one is the primary electron from the electron gun, hits on the gold surface, the gold will emit out the secondary electron. So these secondary electrons will be picked up by the detector and we generate the image. So if you look at the groove, the groove may have more electron. Some part will have less electrons, some part groove may have more electrons. So you're going to see different degree or different intensity of the secondary electrons that can be detected. So the image here will produce a two-dimensional image, but for the scanning electron microscope, we're going to see three-dimensional image. Are you clear? Okay. So let us look at the functions. Transmission electron microscope, generally, we want to study the ultrastructure of a cell. Scanning electron microscope, we want to produce a three-dimensional view of the object, surface view. So how to stain? So we stain it with heavy metal for TEM, for example, uranyl, lead acetates, or osmium tetraoxide. Okay. Then for scanning electron microscope, we coat it with a thin layer of the metal. In this case, we use the gold. So it's very expensive, actually. So what's the principle? Do not need to know, guys. Do not need to know the principle. So basically, the electron beam passed through the ultra thin dehydrated section of the dead specimen, and electrons are absorbed by the heavily stained parts, provide a contrast between the different parts of the specimen. But for scanning electron microscope, the electron beam will hit on the surface of the specimen. So some of the electrons are absorbed and excite other electrons at the surface to give off the secondary electrons. And this electron will transmit it to form a three-dimensional image. Okay. So one thing that you need to know here is to operate this transmission electron microscope is not easy. You need expertise to do it. Okay. Can you see that? Okay. Then what we can use it here is, if you compare both of them, this parts, the chamber here, when you want to operate, you need to operate it in the vacuum. So first thing, you need to suck out the air from the chamber, because if no electron bombard with the air and the dust, then confirm you're going to generate the heat. Are you clear? Clear. So inside here, it must be a vacuum. Okay, so entire operation is not everybody can do it, okay, because you need the expertise to do it, okay. And one electron microscope may cost you, okay, electron microscope may cost you millions, okay. The normal microscope will cost about 4,000 to 5,000. This one millions, a few millions. You want to buy one electron microscope, and you have to operate it in a clean room, okay. So one question here, can we use both gold or only scanning? Normally, we don't use gold for the transmission because it's not so good. Even though it's heavy metal, but the effect is not so good, okay, compared to other metal that manage to block the electrons beam better, okay. So if you compare, last parts, if you compare the light microscope and electron microscope, source of radiation, light microscope, we use a visible light ray. Electron microscope, we use electron beam. Maximum resolutions. Now, when I say the maximum resolution, basically in this case, okay, it depends on the light source, because visible light source, we use the shortest one, 400, that's why 200 nanometer roughly. And electron microscope will be 1 nanometer for TM, 10 nanometer for SEM. Now, even though we can go down to 0.5 nanometer or even shorter, but normally we do not need to be so high in the resolution, because 1 nanometer, generally we can see most of the things already. Because if you increase the, you decrease even more, okay, let's say for example, is use 0.5, energy needed is very high. So you will incur the cost, that's one. Second, if the energy is too high, you're going to pass through your specimen, right? Can you see that? So no point. Okay, so that's why we don't increase further. Okay, two reasons why. 1 nanometer, 10 nanometer, sufficient for us to view the ultra structure or surface structure already. Okay, if we increase it, let's say that the resolution to 0.5 nanometer or 0.1 nanometer, useless, because you won't actually see more detailed thing. Second, you're going to incur more cost because you need to generate higher power. Okay, so maximum magnification that produce clear image about 1,500 times, but electron microscope roughly about 500,000 times. Okay, condenser lenses that we use here, as I say that for light microscope, we use the lens, it's a glass. Electromagnetics or electromagnets, we use it for the electron microscope. Okay. In terms of biological specimen, light microscope, living or dead sample. Electron microscope, dead sample only. It must be dehydrated. Stain, you can use unstained or stained by color dyes. Okay, you can use methylene blue, you can use the iodine to stain your specimen. Electron microscope, we stain it with heavy metal. Preparation of specimen, if you think that light microscope, or when you go back to the lab, we will do this experiment, prepare specimen. You will think that preparing a specimen is so tedious and so difficult. Wait until you process the electron the specimen used for electron microscope, because it's very time consuming. It's not like 1 or 2 days work. You have to prepare the specimen, and the operation require expertise to do it. Okay. Cost, definitely relatively cheaper, small and portable. Nowadays, you can have the use of power bank. If you're portable, you can use this charge microscope, and you can bring it to the jungle forest to study the specimen. But electron microscope, you cannot move them. It's very expensive, large, and operate in a very special room. What means special room? In this case, the room must be a clean room so that not many dust. Okay. So with this, I have done for the chapter 1. Okay, so I stop recording.